Let us proceed to acid secretion, enteric nervous system, vomiting center, and other secretory functions. So for the first topic, it is physiology of acid secretion. The regulation of acid and pepsin secretion reflects the intricate balance of chemotransmitters delivered to the gastric mucosa by several pathways that mediate both stimulatory and inhibitory mechanism. Functional anatomy of the stomach. The stomach consists of three anatomical: fundus, corpus, and antrum, and two functional areas: oxyntic and pyloric glands. The oxyntic area comprises approximately 80% of the stomach and contains parietal cells that produce gastric acid. Also present in the oxyntic glands are neuroendocrine cells, producing paracrine and hormonal agents that modify parietal cell activity. The stomach is divided into four regions: the cardia, which surrounds the opening of the esophagus into the stomach; the fundus of stomach, which is the area above the level of the cardiac orifice; the body of the stomach, which is the largest region of the stomach; and last is the pyloric part, which is divided into the pyloric antrum and pyloric canal, and it is distal end of the stomach. Phases of gastric acid secretion. The physiologic stimulation of acid secretion has classically been divided into three interrelated phases: cephalic, gastric, and intestinal. The cephalic phase is activated by the thought, smell, taste, sight, chewing, fears, and emotional stress. The cephalic phase of gastric secretion is mediated entirely throughout the vagus nerve. It contributes about 30 to 50% of the total postprandial acid production. The vagus nerve is the sole neural link between the brain hierarchy. The gastric phase is due to the chemical effects of food and distension of the stomach. Gastrin appears to be major mediator since the response to food is largely inhibited by blocking gastrin action and its receptors. The gastric phase is period in which swallowed food and semi-digested protein activate gastric activity. The intestinal phase occurs in the duodenum as a response to the arriving chyme, and it moderates gastric activity via hormones and nervous reflex. The duodenum initially enhances gastric secretion but soon inhibits it. The stretching of the duodenum accentuates vagal reflexes that stimulate the stomach, and peptides and amino acids in the chyme stimulate the G cells of the duodenum to secrete more gastrin, which further stimulates the stomach. So now what is chyme? Chyme also stimulates duodenal enteroendocrine cells to release secretin and cholecystokinin. The hormone primarily stimulates the pancreas and gallbladder, but they also suppress gastric secretion and motility. The key player in acid secretion is hydrogen-potassium ATPase or proton pump, located in the canicular membrane. Mechanism of acid secretion. Hydrogen ions are generated within the parietal cell from dissociation of water. The hydroxyl ion formed in this process rapidly combined with the carbon dioxide to form bicarbonate ion, a reaction catalyzed by carbonic anhydrase. Bicarbonate is transported out of the basolateral membrane in exchange for chloride. The outflow of bicarbonate into blood results in the slight elevation of blood pH, known as alkaline tide. This process serves maintain Cellular pH in the parietal cell. Chloride and potassium ions are transported in the lumen of the canalicus by conductance channels, and such necessary for secretion of acid. Hydrogen ion is pumped out of the cell into the lumen in exchange for potassium through the action of the proton pumped. Potassium is thus effectively recycled. Mucosal protective agents. Mucosal protective agents, any drug that protects the mucosal lining of the stomach from acidic gastric juices. The mucosal barriers is the name given to the barriers of the stomachs that resist the back diffusion of hydrogen ions. The barrier is a layer of thick mucus secreted together with an alkaline fluid. Since the mucus is a gel, it entraps the alkaline fluid so that the stomach is coated. Sucralfate and prostaglandin is examples of mucosal protective agents. Sucralfate is a salt of sucrase complex to sulfated aluminum hydroxide. In water or acidic solution, it forms a viscous, tenacious paste that binds selectively to ulcers or erosion for up to 6 hours. Sucralfate has limited solubility. It breaks down into sucrosulfate and aluminum salt. Pharmacodynamics. Negatively charged sucrosulfates bind to positively charged proteins in the base of ulcers or erosions. A physical barrier is formed. Barriers restricts further caustic damage and stimulate mucosal prostaglandin and bicarbonate secretion. Prostaglandin. The human gastrointestinal mucosa synthesizes a number of prostaglandins. The primary ones are prostaglandins E and F. Misoprostol is approved for gastrointestinal conditions. After oral administration, misoprostol is rapidly absorbed and metabolized to a metabolically active free acid. Pharmacodynamics. Misoprostol has both acid inhibitory and mucosal protective properties. It stimulates mucus and bicarbonate secretion and enhances mucosal blood flow.
So in this video, we are going to discuss about the physiology of enteric nervous system. It is important to know that gastrointestinal system has its own nervous system, which is called the enteric nervous system. The enteric nervous system is a quasi-autonomous part of the nervous system and includes a number of neural circuits that control motor functions, local blood flow, mucosal transport and secretions, and modulates immune and endocrine functions. So, ang enteric nervous system or ENS ay tinatawag din na second brain or gut brain. Bakit second brain? Dahil ang ENS ay kayang mag-endorse ng functional activities autonomously or independently, and it can also communicate to central nervous system through parasympathetic and sympathetic nervous system. So meaning, ang enteric nervous system is also influenced by the central nervous system. It is also important in controlling gastrointestinal movements excretion. So the principal components of enteric nervous system are two networks or plexuses of neurons, and both of each are embedded in the wall of the digestive system and extend from esophagus to anus. The first network is mesenteric plexus, which is also called Auerbach's plexus, and the second one is submucosal plexus, also known as Meissner's plexus. So in this picture, it is a sectional of small intestine, and let's see where exactly these plexuses are located in the wall of gastrointestinal tract. So the small intestine is made up of four layers of tissues. The first one is the mucosa. So ito yung mucosa. It forms the inner layer of epithelial tissue. It is specialized for the absorption of nutrients from the chyme or chyme. So the next is the submucosal layer. Ito. It provides blood vessels, lymphatic vessels, and nerves to support the mucosa on the surface. And the third one is the muscularis externa. So it is composed of two layers of smooth muscles. The inner circular muscle layer, ito, and the longitudinal muscle layer. So finally, the fourth one is the serosa, which forms the most layer of epithelial tissue. So balikan natin yung two components ng enteric nervous system. And unahin natin ang mesenteric plexus. So the mesenteric plexus is located at the outer plexus lying between the longitudinal and circular muscle layers. Sa mesenteric plexus ay nasa pagitan ng circular muscle layer at longitudinal muscle layer. So ito siya, yung tinuturo ng arrow. The mesenteric plexus is consists mostly of a linear chain of many interconnecting neurons. Ito yung mesenteric plexus. That extends the entire length of the gastrointestinal tract. It also controls mainly the gastrointestinal movements. The functions of mesenteric plexus when stimulated are the following. It increases the tone of the gut wall. It also increases the intensity of the rhythmical contractions, and it slightly increases rate of the rhythm of contraction. And also, it increases the velocity of conduction of excitatory waves along the gut wall, causing more rapid movement of the gut peristaltic waves. So the second network of enteric nervous system is the submucosal plexus. It is in the inner plexus which lies in the submucosa. So ito yung submucosal plexus. It concerned mainly with controlling function within the inner wall of each minute segment of the intestine. The submucosal or Meissner's plexus is involved with the local conditions and controls local secretion and blood flow to the submucosa, which of course determines absorption. So, kinokontrol ng submucosal plexus ang mga local muscle movements. So, in general, the enteric nervous system is consists of about 100 million neurons, nearly equal to the number in the entire spinal cord. So, let's discuss kung paano gumagana ang enteric nervous system kapag tayo ay kumakain or kumain. I'm going to explain first how the ANS controls the peristalsis, and it is also important to know how the ANS works into two different ways. So first, it functions autonomously using the local reflex or the autonomic local reflex. And second, it also functions in communication with sympathetic and parasympathetic nervous system. So, paano nga ba gumagana ang autonomic or local reflex ng ANS? So the picture here shows a sectional small intestine somewhere between the duodenum and ileum. So makikita natin yung four layers ng tissue ng small intestine, which is the mucosa. Ito yung mucosa. Circular muscle layer, ito. The longitudinal muscle layer. At ang huli ay ang serosa. And over here is the mesenteric plexus, which consists mainly of two types of neurons, the ascending network of neurons and the descending network of neurons. Meron ding two types of sensory neurons. So the first one here receive the stimulus. Ito, yung una na sensory neuron. It receives the stimulus from the smooth muscles. At ang pangalawa ay nakolocate dito sa submucosa. So in this part, sa part ng submucosa, makikita ang submucosal plexus. So ito ang submucosal plexus. Ayan. So for example, when you eat a burger or a hamburger and a bolus has reached the small intestine, so a bolus is a soft mass of chewed food. So from here sa mouth or oral, the bolus it will travel toward the aboral part. Then in the intestine, the bolus distance and stretches the intestinal smooth muscles. Then the intestinal distension further stimulates. So ito, the intestinal distension further stimulates the sensory neurons ng mesenteric plexus. Also, this sensory neuron, itong nasa submucosa, will stimulate by chemical composition of the food. Both the sensory neurons give branches to the ascending and descending interconnected neuron and stimulated that. So after my stimulate, mag-release ang ascending network of neurons dito. Mag-release ang ascending network of neurons ng excitatory neurotransmitters gaya ng acetylcholine at substance P sa circular muscle layer behind the bolus that causes contraction of the circular muscle layer behind the bolus. And in the same time, the descending network neuron, ito ang descending network neuron, will release an inhibitory neurotransmitter. So mag-release ang descending network neuron ng inhibitory neurotransmitters gaya ng nitric oxide at VIP or vasoactive intestinal peptide na magiging dahilan ng relaxation ng muscle or ng relaxation ng longitudinal muscle. But in front of the bolus, it needs to have a relaxation of the circular muscle along the bolus to move forward. So dito naman tayo. And therefore, in front of the bolus, ito, in front of the bolus, mag-release ang descending neuron ng inhibitory neurotransmitters gaya ng nitric oxide at vasoactive intestinal peptide. Ayan. Para mag-cause ng relaxation ng muscles. So, in the same time, or at the same time, mag-release din ang ascending neuron. Ito, mag-release ang ascending neuron, itong part na to, ng excitatory neurotransmitters like acetylcholine at substance P sa longitudinal muscle na magkukos ng contraction ng muscles. So to sum it up, for the behind the bolus, the circular muscles will contract and the longitudinal muscle will relax. Whereas in front of the bolus, the circular muscles will relax and the longitudinal muscles will contract. So that is for the autonomic or local reflex of enteric nervous system. Punta naman tayo sa parasympathetic at sympathetic nervous system. As I have mentioned earlier, the ENS also functions in communication with both parasympathetic and sympathetic nervous system. The sympathetic neurons influence in aeration to both plexuses supplying my enteric plexus and submucosal plexus. It is almost the same for parasympathetic nervous system, but they work opposite to each other. So the sympathetic nervous system stimulates the body's fight or flight response. It will be activated in case of threat, stress, or fear. And those kind of situation doesn't need to increase gastrointestinal activity because you have to focus on your emotion or on your situations because you are experiencing threat, stress, and fear. So therefore, the sympathetic nervous system inhibits both plexuses, decreasing peristalsis, blood flow, absorption, and local secretion in mucosa. So the stimulation of the sympathetic nervous system inhibits activity of the gastrointestinal tract, causing many effects opposite to those of the parasympathetic system. So it exerts in effects in two ways. First one is to slight extent by direct effect of secreted norepinephrine to inhibit intestinal tract smooth muscle, except for the mucosal muscle it which it excites. And the second one is to a major extent by an inhibitory effect of norepinephrine on the neurons of the entire enteric nervous system. So the strong stimulation of the sympathetic system can inhibit motor movements of the gut so greatly that this can literally block movement of food through the gastrointestinal tract. As discussed from the previous module, the parasympathetic nervous system is responsible for the rest and digest response. For example, when you drink a milk, the parasympathetic nervous system will be activated and dominates and will stimulate both plexuses. The stimulation of parasympathetic increases activity of the entire nervous system. The parasympathetic supply to the gut is divided into cranial and sacral divisions. Except for a few parasympathetic fibers to the mouth and pharyngeal regions of the alimentary tract, the cranial parasympathetic nerve fibers are almost entirely in the vagus nerves. These fibers provide extensive innervation to the esophagus, stomach, and pancreas, and somewhat less to the intestines down through the first half of the large intestine. So the parasympathetic nervous system increases the peristalsis, blood flow, absorption, and local secretion in mucosa. Let's go now to the gastrointestinal reflexes. The anatomical arrangement of the enteric nervous system and its connection with the sympathetic and parasympathetic system supports three types of gastrointestinal reflexes that are essential to gastrointestinal control. The three types of gastrointestinal reflexes are the following. The first one is the reflexes that are integrated entirely within the gut wall enteric nervous system. So these reflexes include those that control much gastrointestinal secretion, peristalsis, mixing contractions, local inhibitory effects, and so forth. Second one, it reflexes from the gut to the prevertebral prevertebral sympathetic ganglia, and then back to the gastrointestinal tract. So itong reflexes na to ay magta-transmit ng signal sa mga malalayong distansya papunta sa mga areas ng gastrointestinal tract, such as the signals from the stomach to cause evacuation of the colon or the gastrocolic reflex, at signals na galing sa colon at small intestine para mag-inhibit ng stomach motility at stomach secretion or the enterogastric reflexes and the reflexes from the colon. So the third reflex or the third gastrointestinal reflex is the reflexes from the gut to the spinal cord or brainstem and then back to the gastrointestinal tract. So these reflexes include especially the reflexes from the stomach and the duodenum to the brainstem and back to the stomach by way of the vagus nerves to control gastric motor and secretory activity. The other one is pain reflexes that cause general inhibition of the entire gastrointestinal tract, and the third one is the defecation reflexes that travel from the colon and rectum, the spinal cord, and back again to produce the powerful colonic, rectal, and abdominal contractions required for defecation or the defecation reflexes. So that is all for the physiology of entire nervous system.
So now let's proceed to other secretory functions of alimentary tract. Pancreatic secretion. The exocrine pancreas secretes an aqueous mixture into the duodenum composed of digestive enzymes which aid in further degradation in ingested food and bicarbonate which helps neutralize stomach acid. Neutralization of gastric acid is a critical function of the pancreas as the small intestine mucosa is a specialist for nutrients absorption and thus cannot possess a thick protective mucous layer similar to that of the stomach. Furthermore, pancreatic digestive enzymes are optimally active at basis pH, and thus pancreatic bicarbonate secretion is a key requirement for the proper digestion. Bicarbonate secretion. Pancreatic bicarbonate secretion is essentially the reverse of stomach acid secretion and primarily occurs in pancreatic ductal cells. Here, carbon dioxide is combined with water to form carbonic acid subsequently. The carbonate ion is transported into the pancreatic duct, while the hydrogen ion is transported into the bloodstream. Molecular mechanisms. Carbon dioxide within pancreatic ductal cells is transformed by carbonic anhydrase to carbonic acid, which immediately splits into hydrogen and bicarbonate. The generated hydrogen ion is secondarily actively transported into the bloodstream. Digestive enzyme secretion. Pancreatic enzymes are synthesized and stored in a pancreatic acinar cells as inactive zymogens. As a result, these powerful digestive enzymes are prevented from destroying pancreatic tissues. As described, the enzymes only become active once fully secreted into the body of the small intestine, which possesses sufficient mucosal protection. Pancreatic enzymes. The exocrine pancreas secretes a variety of digestive enzymes specialized for degradation of all the different basis biological macromolecules. Regulation. Pancreatic secretions are primarily regulated by the three factors which act synergistically to potentiate one another's effect. First is acetylcholine, released by vagal efferents, primarily stimulates synthesis of digestive enzymes by pancreatic acinar cells. 2. Cholecystokinin is released by eye cells in the duodenum and jejunum upon entry of food and primarily stimulates synthesis of digestive enzymes by pancreatic acinar cells. And last, secretin is released by S cells of duodenum in response to entry of low pH stomach acid and primarily stimulates production of aqueous sodium bicarbonate solution by pancreatic ductal cells. Phases of pancreatic secretion. First is cephalic phase. Phase is initiated by sensory experience of seeing and eating food and primarily involves vagus nerve stimulation of acinar cells to produce digestive enzymes. Next is gastric phase. This phase is initiated by the presence of food within the stomach and is once again primarily involves vagus nerve stimulation of acinar cells to produce digestive enzymes. And last is intestinal phase. Phase is initiated by emptying of stomach contents into the small intestine and involves release of both secretin and cholecystokinin, which stimulate pancreatic ductal cells to synthesize aqueous sodium bicarbonate solution. Bile secretion. Bile is a bitter tasting, dark green to yellowish brown fluid produced by the liver. Bile aids the process of digestion of lipids in the small intestine. Bile is stored in the gallbladder and upon eating is discharged into the duodenum. Bile is composed of the following materials: 85% of water, 10% of bile salts, 3% of pigments, 1% fats, 0.7% inorganic salts, and 0.3 cholesterol. Secretion occurs in three stages. One is formation of secretion of constituent of bile by the liver. Bile salts are synthesized in hepatocytes and transferred into bile canaliculi. Bile salts, along with glucose, amino acids, and bile pigments, are actively secreted into canaliculi. Next is filling of gallbladder. Bile then passes through the biliary tree and ultimately to the hepatic duct, and then it reaches the gallbladder where it is concentrated and stored temporarily. And last is discharge of bile in the intestine. Enterohepatic circulation is a continuous circulation of bile salts from liver to bile enter passage into the duodenum, their absorption in the ileum, and then return to the liver as mixture of bile acids and salts. Regulation of bile in the liver. Bile acid dependent means bile secretion increases as more and more bile acids are presented to the liver by enterohepatic circulation. It occurs after fatty meal. When intestine is empty, this mechanism stops. Bile acid independent naman means it is mainly due to transfer of electrolytes by pumps. It is under the influence of secretin. Functions of biles. First, digestive. Helps in digestion of fats. The next is absorptive. Helps in absorption of fat-soluble vitamins, iron, and calcium. And then third is laxative. It stimulates peristalsis movements of intestine and thereby helps in defecations. Fourth is chlorhydric, helps in secretion of bile from liver. And last is excretory. Certain substances like heavy meals, certain drugs, bile pigments, and excess cholesterol. And bile acts as lubricant for kind in intestine.